Welcome to the mPrep system reagent processing instructional video. This video will show how easy and efficient reagent processing is using the mPrep system for preparation of TEM and SEM specimens. mPrep S specimen capsules are easily loaded directly from the mPrep workstation. Capsules can also be loaded individually. Have the reagents ready along with several solvent-resistant reservoirs. Simply pour a small volume of the first reagent into the reagent reservoir. Set the reagent aside. Ensure that the mPrep S capsules are attached. mPrep F couplers are used to ensure that the reagent is not accidentally drawn into the pipetter. Draw the first reagent into the pipetter. This process is called aspiration. It's very convenient to rest the pipetter using a lab stand during reagent treatments. Let the reaction proceed per your protocol for your sample preparation process, and when you are ready, pour the second reagent, such as a buffer, into a clean second reservoir. Then, take the pipetter and expel the first reagent. A gentle touch of the capsule tips to the reservoir will remove droplets from the ends of the capsules. Next, slowly aspirate in the second reagent. Rest the pipetter on the laboratory stand and let this reaction proceed. Simply repeat this process for all reagent exchanges. Multiple exchanges of the same reagent can be done from the same reservoir, such as several rinses or to provide reagent agitation. Proceed per protocol for all reagents, such as fixatives, rinses, stains, dehydrants, and resin infiltration. Of course, for SEM sample processing, there is no resin infiltration. After solvent dehydration, the capsules can be transferred to the mPrep critical point dryer holder and then processed through the critical point, or may be processed through other SEM sample preparation protocols. After the specimens are entirely dehydrated, begin the infiltration process with resin. This step shows the first resin infiltration with 100% resin. It is important to set the pipetter for a resin volume of 100 to 150 microliters of resin to provide a sufficiently large block. These larger volumes are also easier to handle. After first expelling the solvent, such as acetone, slowly aspirate the resin into the capsules. A slow aspiration is necessary to prevent bubbles forming in the viscous resin. The pipetter is then left standing in the reagent reservoir for the appropriate infiltration time, which may be overnight. After the infiltration is completed per protocol, the resin in the capsules is then ready for curing. The capsules are first firmly inserted into the mPrep silicone bench, and then the capsules are ejected. Note, the aspirate/dispense button on the pipette is not touched. As shown, the mPrep F couplers can be removed and reused if they are not contaminated. It is not necessary to remove the couplers before curing, simply transfer directly to the oven with the couplers still attached. The couplers can then be removed after curing. If a larger, longer block is desired, after removing the couplers, simply top off each capsule with more resin. This is an excellent time to insert labels for embedding, as it is standard practice in many labs. The filled capsules in the mPrep bench are then placed into an oven for curing, per your protocol. The cured blocks are now ready for sectioning. Later videos will show how easily this is done. The reagents can be arranged using many different methods. Individual reservoirs were just shown. It is also possible to lay out reagents using 12 well plates, or 96 well plates, or in combinations, depending on the protocol. Here, we see a 96 well plate, a 12 well plate with labeled wells, and reservoirs on the lab stand. 12 well plates are quite convenient. Small amounts can be placed in each individual well, and it's very easy to step through the process using a single well plate, as you can see in this demonstration. Note that the timing is just for demonstration purposes. This method of exchanging reagents with multiple pipetters, reservoirs, and well plates may be used for any type of processing with mPrep specimen capsules or with mPrep G capsules for grid processing. Thank you for watching. If you would like further information, please contact Microscopy Innovations at info@microscopyinnovations.com.